Hey AP biologists, it's Mrs. Coleman here. This video is all about gene expression in eukaryotes. So what gene expression means is how we get from the information stored in DNA, how do we get that information to actually build a functional protein product? And so if you take a look at your notes sheet, we'll start by filling in the definition for gene expression. And so here it is. Gene expression is the process by which information from the gene is used to create a functional gene product. And the picture on the bottom of the screen here kind of gives you an idea of what that process looks like. We've got the information encoded in our DNA, in our genome, and that information is then rewritten into a different code called messenger RNA, and then that messenger RNA is used to build a protein that can function in the body doing a number of things. Maybe it's an enzyme or an antibody, etc. And so when we talk about gene expression, we're kind of figuring out where do we control this process? How can we increase the speed at which proteins are produced? How can we turn the gene expression process off if we don't need those proteins in a given cell or at a given time? And when we look at the process of gene expression, there are a lot of different steps. We have to first get from our DNA to make RNA. Our RNA has to mature so it can be used. It has to get transported out of the nucleus to the ribosomes to be made and to be used to make those proteins, and then we have to process those proteins before they can become functional. And what that really means is that we can turn this process on or off, or speed up this process or slow this process down at a bunch of different steps along this process. And so when you look at your notes, you can see the 5 places that we can turn the process on or off, or speed it up or slow it down. The first place is at the genome level. So here we're talking about the chromosomal level, and we start with our chromatin, and then we have our gene that's available to be expressed. We can control how quickly the transcription of that gene occurs into pre-messenger RNA. We can control how that pre-messenger RNA is processed to create messenger RNA that's actually usable. We can then control how that messenger RNA is used to build proteins, and we can also control how those proteins are then processed to become functional units. And so we have 5 places throughout the process where we can control the expression of these genes. And so we're going to talk about each of these different parts, and I'll stop along the way and make sure that you know where to fill the stuff in on your notes sheet. So let's start with the genomic regulation of gene expression. Reminder, chromatin, that's the complex of DNA and proteins that forms within the chromosomes in the nucleus of a eukaryotic cell. So here we've got our DNA, it gets wrapped up around these histone proteins, and then those get wrapped up on top of each other to create our chromosomes. If you want to, you may jot down the definition of chromatin right next to the word chromatin on your notes sheet. But if you remember this definition from previous units, you can go ahead and skip writing that down. This is something you should write down underneath where it says DNA methylation on your notes. So methyl groups, which are CH3s, you can see them here, if they get added to the DNA strand, that actually blocks the transcription factors from binding. Remember, transcription factors are going to bind to the DNA and help the RNA polymerase bind to the promoter to actually transcribe the gene. But if these methyl groups are attached to the DNA, those transcription factors can't bind, which means that the gene expression is inhibited. We're not going to produce messenger RNA, and if we don't produce messenger RNA, we are not producing a protein. So methylation is one way that we can decrease gene expression. There's another process called histone acetylation, and this is where we add acetyl groups here to our histones, the proteins, and what that's going to do is it's going to loosen up the chromatin. So here you can see it's really tightly bound around the histones. Once we've added those acetyl groups, the DNA kind of uncoils a little bit, and now we can see that there's some space for us to access the DNA. So our transcription factors can bind, the RNA polymerase can bind, and therefore it's going to promote gene expression. So right away in our chromosomes, in our chromatin, we're going to see that we have ways to increase or decrease gene expression. So let's say that we have our chromatin and we have our gene that's now available for expression. So the chromatin has loosened up, we don't have a lot of methylation, we're able to attach our transcription factors and our or RNA polymerase. So let's talk about the ways that we can effectively control the process of transcription. The first is with these transcription factors. We've talked about these a little bit before. Transcription factors are going to initiate transcription by binding to the promoter. Remember, the promoter is like kind of that like start signal in our DNA. And once those the transcription factors bind, RNA polymerase is able to bind and transcription can begin. So if we have a lot of transcription factors, we're going to have lots of transcription occurring. If we have less transcription factors, less transcription will occur. And that's how we can regulate gene expression in our cells. We also have these things called enhancers. These are sequences that are very far from the gene. So if the gene is over here and the promoter is over here, our enhancers are upstream of those genes. And activators can bind to them, and when that occurs, the DNA bends. We get our transcription factors and our mediator proteins that bind, which allows our RNA polymerase to bind and RNA synthesis to begin. So this is going to increase our possibility of creating our functional protein products, increasing gene expression. We also have things that are called repressor proteins. These are similar to the repressor proteins that you learned about in the operon system. They're going to bind to the DNA and prevent the RNA polymerase from binding, which ultimately blocks transcription. We're not going to create messenger RNA. We're not going to make any proteins. The gene is not expressed. So let's say we've got our chromatin, we've made our gene available to express it, and we've actually created a strand of messenger RNA. But that strand of messenger RNA is not able to be used yet. There's lots of pieces of information within that messenger RNA that are just not usable. And so we have to cut them out. And so we talked about RNA processing in our notes in previous classes. But what can also happen is that we can have what's called alternative RNA splicing. And that's when we treat exons and introns differently depending on what kind of protein products we're making. So here we have one gene, and we have these exons which we're going to keep, and the introns here in these blue sections that we're going to cut out. So when we make that primary RNA piece, we have all of the exons and all of the introns still in place. But if I cut out the introns and some of the exons, I can make this strand of messenger RNA. And take a look. Here's the protein product that I create. But I can cut this piece of messenger RNA differently. Let's say I cut out the introns plus exon 2 and exon 4. Well, now this is the functional messenger RNA, and this becomes my functional protein product. And you can see this protein and this protein have different structures, and therefore they're going to have different functions. And this alternative RNA splicing kind of explains why we have fewer genes than actual proteins. We've got about 10,000 different genes in our genome, but we can make between 20 and 30,000 different proteins, and those numbers just don't quite add up. But that's because each gene actually codes for up to several different kinds of proteins depending on which pieces we treat as exons and which pieces we treat as introns. And so we want to process that messenger RNA, and that process is going to kind of change the way those genes are expressed in the end. So another way we can control gene expression. So once I have a functional piece of messenger RNA, we transport it out into the cytoplasm, where it's going to be used to translate into proteins. So over here is where you're going to write the next piece of information. And we also have these things called regulatory proteins, and these are going to help with the initiation of translation. There's going to be these proteins that help the messenger RNA bind with the ribosomes. When we have a lot of these enhancing type regulatory proteins, we're going to make a lot of those protein products in the end. We're going to increase gene expression. However, we can also have regulatory proteins that prevent the messenger RNA and the ribosomes from attaching. In that case, we're not going to make functional protein products with the ribosomes, and the gene won't be expressed. So those regulatory proteins are going to help to control the proteins that we are making in the expression of the gene. Now, once we've created our proteins, our messenger RNA is going to get broken down, and the pieces, the nucleotides, can be recycled to make new messenger RNA. As long as we have messenger RNA kind of floating around in the cytoplasm, we're going to make proteins out of it. So our cells are going to break it down when we are no longer in need of making more proteins. And so messenger RNA degradation, the breakdown of messenger RNA, is going to be one way that we can also turn gene expression off. And the lifespan of that messenger RNA is determined by the caps that we talked about in our discussion of messenger RNA. We've got this modified guanine cap on the 5' end. We've got this poly A tail, a bunch of those adenines repeating over and over again on the 3' end. And those caps are going to kind of determine how long that RNA lives in your cytoplasm, and therefore how long the gene is being used to make those proteins. And so you can jot down this information kind of on that little arrow that slides off here to the left. Messenger RNA degradation goes over here. So let's say we've used our messenger RNA to actually create a polypeptide. Just as when we made our first strand of messenger RNA, that messenger RNA wasn't ready to be actually used. Same thing here. Once we first build that polypeptide chain, it isn't ready to be used. It's not a functional protein product. And so we have to do a number of things to get it ready to be a functional protein product. And it's not until that protein is functioning that we say that the gene is being truly expressed. So we've got these different types of protein processing. First, some proteins have some extra amino acids in them. For example, insulin is a really good example of this. We have a pre-insulin and an active insulin. When we first make pre-insulin, there's some extra pieces of, extra sequences of amino acids that we actually cleave or cut out. And then once those pieces have been cleaved off, the insulin is actually active. So in some of our proteins, we have to remove portions of the polypeptide in order to activate and have a functional protein. We also might add some different chemicals to our proteins, maybe lipids or carbs or other functional groups. Hemoglobin is a good example of this. Our hemoglobin carries oxygen in our red blood cells, and there's heme groups, ions, attached to it, and so those hemoglobin molecules are not going to be functioning until they have those heme groups attached. So that's a chemical modification that we have to make in order for our genes to be truly expressed. We also have to fold those amino acid chains into their secondary and tertiary structures, and so we have some proteins that are going to help to ensure that the proteins are being folded in the appropriate manner so that they have the right three-dimensional structure and therefore the correct function. We also have to transport the proteins to their site of activity. So for example, again, if I use insulin, just making insulin and storing it in your cells isn't actually a functional gene product because the insulin is just sitting there. It's not doing anything. So until that insulin is released fully into your bloodstream, the gene is technically not fully expressed at that point. So here we can see a lot of different examples. This is actually the pre-insulin, pre-proinsulin, insulin, and then here we have to actually release it out into your cells, and it's not until that insulin gets released that the gene is being fully expressed. Now, we also want to break down our proteins, and that's going to ultimately end the expression of that gene. In order to break down our proteins, we'll mark them with another molecule called ubiquitin, which kind of calls over an enzyme that destroys proteins called proteasomes, and the proteasome is going to break it down into the little protein fragments, little peptide chunks, and we can actually reuse those peptides, those amino acids, to build new proteins. And so here's another way at the very, very end that we can shut down gene expression by breaking down the proteins that we once created. So to kind of recap everything that we've talked about, in order to get a functional active protein, there's a lot of things that have to happen beforehand. We have to use our chromatin and open it up to access our genes. We have to transcribe it into our pre-mRNA. We have to process it so it's a mature piece of messenger RNA. That messenger RNA has to be released out into the cytoplasm, attached to the ribosome to create the polypeptide, and then we have to fold it or add things to it or activate it to create that active protein. And so we have a lot of ways that we can turn the process on or off at any of these different steps. There's also ways that we can speed up or slow down these processes, which ultimately are going to regulate or control the expression of genes in our eukaryotic cells. So I hope you thought this was helpful. Go back and take a look at any parts that you need to help you fill out your note sheets, and we'll talk about this again soon.